بسم الله الرحمن الرحيم. في البداية أحب أتوجه بالشكر لأسرة كلية الصيدلة جامعة أسيوط على الدعوة الكريمة، والمحاضرة هتكون عن Dihydrophyllate Reductase as anti-tumor molecular target. As we all know, we have two types of tumors, either controlled or uncontrolled. The controlled, called the benign tumor, while the uncontrolled is malignant tumor. Cancer or the malignant tumor thing is the second leading cause of death worldwide. One of the routes to combat cancer is an enzyme called dihydrophyllate reductase. The dihydrophyllate reductase mainly involved in the production of thymine out of uracil, a component of nucleic acid. Dihydrophyllate reductase, we have quite number of dihydrophyllate reductase inhibitors, but they suffer from toxicity and drug resistance. So there is always a need to increase the selectivity and lower the toxicity of the dihydrophyllate reductase inhibitor. What dihydrophyllate precisely do, as you can see, it reduces dihydrophyllate into tetrahydrophyllate, and a process will involve eventually of the methylation of uracil into thymine. In this scheme, you found the sites which drugs could be involved. We have two sites to prevent the formation of tetrahydrofolic acid. For quite some time, research has been involved. The prototype or the classical inhibitor is methotrexate, because it's called the classical because it resembles folic acid in structure. We have also a bunch of non-classical dihydrophyllate reductase like trimethoprim, methotrexate, and pyritrexate. In our previous effort since 2006, we managed to allocate some potential dihydrophyllate reductase with IC50 around 4 micromole. As you see, there is a resemblance between those drugs and methotrexate by this N-methyl, as you see. We decided to introduce unsaturation on the kinezoline ring in various types, and we managed to allocate these three compounds in 2010 to get IC50 around 4 micromole. In another study, we introduced the small molecule of thiazole in a thioether linkage to the kinezoline, and we started to see improved activity by the allocation of compound 8 at 0.3 micromole. We have to put in mind that methotrexate shows IC50 with 0.08 micromole. So there is still a long way to go. From those previous three studies, we managed to come up with a general recommendation about the structure-activity relationship. The carbonyl group is essential. The basic nitrogen is essential. The bi-electron system and their interspatial distances is essential for activity. Those type of compounds always in the enzyme bucket attached itself to arginine and lysine residues of amino acid. In this study, we have decided to look from other dimension. We start to look at the functional group needed for anti-tumor activity, such as thioether, amide, azo, and thiorido, and we have decided to combine them. Those structures are active anti-tumor agents from the literature. So we have decided to combine them with the DHFR inhibition activity of quinazoline by suggesting these three series. In series A, we combined the thioether from the previous active compounds, as you see, and the amide from the previous anti-tumor agents to get series A. In series B, we kept the thioether methyl sulfide or methyl mercaptide. We kept it as it is, and we added an azo function. And here we added a thiorido function. Now we have a combination of structural features of active antitumors and the structural features of active dihydrophyllate reductase inhibitors. In order to realize this, we managed to synthesize those compounds through traditional schematic schemes. I'm not gonna involve you in the details, but starting with antranilic acid derivatives, we managed to get the nitroquinazoline derivative and then managed to reduce this nitro into amino. Here we start. The amino group has been used to form amide. Various amides, as you see, aliphatic, heterocyclic, and aromatic. Here, we used the amino group to get azo function at position 6, as you see. Also, we use the amino function to get a thiorido functional group into the position designed to be. We took all of this to biological testing. We used the bovine liver dihydrophyllate reductase, which is 75% resemblance to human dihydrophyllate reductase. We used methotrexate as a control, and we did manage to allocate those 4 compounds with biological activity, inhibition of DHFR between 0.2 to 0.3 micromole. We took the compounds also to test it against 4 different types of tumors: colorectal, prostate, epithelioid carcinoma, and breast cancer. And we make an average IC50 to allocate 3 different active compounds, as you see, with IC50 ranged between 6.7 to 9.1 micromole. In general, less than 10 micromole an IC50. As a trial to reveal the mode of anti-tumor activity, we decided to make cell cycle analysis with the most active compound 109 and 111. 109 gave average 10.6 micromole, while 111 gave us 5.5 micromole. 109 in the cell cycle analysis showed apoptotic behavior at the pre-G1 stage. Oh, I'm sorry. and inhibit the cell growth at G2M. As you see, this colored beaks represents it. The same was observed with 111, and it gave the same mode of action, pre-G1 apoptosis and inhibition of cell growth at G2M. We took all of this to molecular modeling. We made the standard conformational analysis of methotrexate, the most active and the most inactive. We made the docking and counted the protein residue involved in that. We also made the docking alignment, the docking experiment in the bucket, and we noticed that the inactive compound is protruding out the bucket, exposing out of the bucket. This is a reason for its inactivity. We made the usual flexible alignment. We found all active compound aligned together, all inactive compound aligned together, and we saw the chaosity when we aligned the active and the inactive together. We made also the surface mapping, and we did notice the resemblance of the shape between methotrexate and the active compound and the deviation of the inactive compound. As a conclusion and the structure activity relationship, we got 4 compounds active as DHFR inhibitors. Also, we got 3 compounds active as anti-tumor agents. When we looked at the structural variation, we did found that the paramethoxyphenyl at the 3-nitrogen of quinazoline contribute much more than the 3-methyl to the anti-tumor activity and the DHFR activity. Compound 79 involved in the bucket through binding with threonine and serine and serine 118 in the same fashion as methotrexate do. We interpreted the inactivity of compound 98 as the S-methyl group protruded, exposed out of the bucket, as you see, and this is why it is poorly active. So we also noticed that compound 109 and 111 exert their anti-tumor activity through inhibition of dihydrophyllate reductase inhibition. The others has some other different mechanism. Also, those 2 compounds exert this activity by dual mechanism, apoptotic behavior and DHFR inhibition. We managed to publish these results in Bioorganic Chemistry last year with those great cooperative people in my group. And I would like here to show you something from Mansoura University. This is our NMR unit, and those are the great people operating this machine and interpreting it. Thank you so much, and I do appreciate being with you in Assiut University Faculty of Pharmacy. Thank you all.
بسم الله والحمد لله والصلاة والسلام على رسول الله. السلام عليكم ورحمة الله وبركاته. Good day, everyone. I hope that all of you are doing fine in the face of this pandemic. First, I would like to express my sincere thanks to the College of Pharmacy of Assiut University and the organizing committee for their kind invitation to this conference. I hope that you will find this presentation useful. The quality of medicine in the market was and still a very hot issue. Counterfeit and low quality medicines have become a global issue because of the continuing growth in the market for these products, especially the counterfeit products, and because of the consequences of their use. Although developing countries are the principal target for counterfeiters, developed countries face many of the same risks also. And sadly, essential medicines like the antimicrobials in general, antibiotics, the antiparasitics, and anti-malarials maybe are the most frequently targeted products of this type in developing countries. Unfortunately, on the other hand, studies with the sound and reproducible methodology on the quality of medicines in developing countries are limited. And convenience sampling is widely used for this purpose, although bias is clearly introduced because usually only the accessible pharmacies or drug selling outlets in these countries are selected. For this reason, there is always a need for an economic probability sampling method which is valid, reliable, and reproducible, and this is what I'm going to talk about in this presentation. So I'm trying to jump to the next slide. I just here declares that I have no conflict of interest. First of all, we should define our objectives. Why do we need to survey? Do we have a problem, or we are anticipating a problem? And also, when do authorities need to survey? Shall every country, every authority in every country regularly survey the market for the presence of low quality medicines or counterfeit medicines? And they should do it routinely, or they should just start when there is a problem. This it depends actually on the country, on the resources. Of course, poor countries cannot do all these things in anticipation. They usually start when there's a problem. And where to survey? In which geographic area of the country, in which city, or in which district, and what kind of pharmacies or drug selling outlets should we survey? Also, this should be in mind before conducting a survey for the quality of medicine. And more importantly, also what to survey. Shall we survey all the market, all the medications available, or only some targeted medication? This again depends on the budget we have. Of course, in some rich countries, they can survey for everything, but in poor countries, again, they may not have the facilities and the resources to survey everything. And how to survey, what sampling method should we adopt? And this is what I'm going to discuss in details. And finally, what are the requirements regarding the time, regarding the cost, the labor also? This is an important issue because it really matters. And again, unfortunately, in some poor countries, the resources are not there, so the quality surveys are limited, and as I mentioned, and they actually face lots and lots of financial troubles with that. Regarding the selection here, so we have to select the area to be sampled. We have to select the medicines to be surveyed. We have also to select the sampling sites. What are these sampling sites? Pharmacies or other outlets. In some countries, even groceries can sell prescription medicines, and maybe these days they also may sell some fake anti-COVID-19 medications. there should be a very flexible and sound sampling design, actually, especially in these low-income countries or poor countries. Regarding the sampling plans, how many dosage units, how many tablets, capsules, vials, or bottles should we sample? If we refer to the pharmacopoeia, like the US pharmacopoeia, the British or the European, the Japanese, for the content analysis and content uniformity, usually they require a large number of dosage units, ranging from 20 up to 60 or sometimes 80 units. The unit used. And again, regarding the sample collection, is overt sampling useful, or shall we go for the simulated client or mystery shopper technique? This again, it depends on the situation and the country. Finally, we have to think about the storage and transportation of the samples. Sometimes the on-site testing for the samples is not available. So we have to send the sample to a lab. So we have to make sure that the transportation facilities is air conditioned and those samples are kept within reasonable temperature and humidity. And finally, the testing itself, what technique we are going to use. Of course, these days, there are some techniques which are based on X-ray or infrared. They can be used on site. But again, in some poor countries, maybe these facilities are not available. So we should go for the lab analysis. I'm going to talk briefly about different sampling methods. When I say sampling here, I mean sampling for the pharmacies. So the sample here is the pharmacy, not the medicine. Most of this published studies regarding the quality of medicines use convenient sampling because it is very simple. But on the other hand, it's a non-probability sampling technique. We don't need to have the full list of pharmacies in an area. We just start randomly in any street, in any district, and we just go at pharmacy or drug selling outlet. So it is prone to bias, this method, because we are just focusing on one area. This area may be the source of the problem, or it's maybe free of the problem, so we don't know. And this is where the bias gets. So maybe this method actually it may lead to false negative results, and one should be very careful when using this sampling method. And finally, we cannot generalize these results because, again, we are stuck to one district or one geographic area. So it may be suitable for surveying high-risk areas by the authorities like the Sentinel monitoring, for example. The second method, which is the very famous one, the simple random sampling, it is a probability sampling technique. It is a true random sampling method, but here we need the full list of pharmacies, and it requires actually a larger sample size of the outlet. Some troubles when deciding the sample size. But this method is reliable, it is generalizable, it can give an accurate estimation of the prevalence of high-risk areas or high-risk sites or geographic regions, or the high-risk also drug selling outlets. Are those pharmacies within the private sector or the public, the government? Unfortunately, this method is very costly, requires time, requires labor, and I'm sure we all know this fact. Another form of the random sampling is the stratified random sampling. Here the outlets are divided into some strata or some layers. Again, we can study the public sector, the government sector, urban areas versus rural areas. We can divide the countryside, the whole country into geographic regions, or the whole city into several districts, and then we have to sample from every district. I mean random sampling, of course. So we have to adjust for the sample size according to the proportionality of the pharmacies within each stratum, each layer, each district, whatever our definition is. It has a comparable advantages and disadvantages to the simple random sampling, of course. Then we have the sentinel site monitoring. This is actually used by the authorities. They monitor the quality of medicine in a specific area in the city where there's always a problem, where counterfeit or low quality medicine are found. Here in the sentinel site monitoring, we can use various selection techniques. They can go for convenience sampling or simple random or whatever. But the owners of these pharmacies may be aware by time they realize that they are being watched by the authorities. So this will lead to a change in the behavior. So they may hide actually the counterfeit products when they expect a sudden campaign by the authorities. Finally, I'm going to talk about lot quality assurance sampling as a recommended first step sampling method for the quality of medicine. Actually, LQAS, or lot quality assurance sampling, as we can see, it was developed in the 1920s. This sampling method was used to determine the sampling size in industry. Let's imagine anything, glasses, for example. If the manufacturer just finished a lot or a batch of eyeglasses this day, maybe 10,000 glasses, they probably need to check for the quality of the product. So they have to select random sample, and the sample will undergo quality analysis, quality testing. After that, the manufacturer will decide that whether this lot is good for marketing or not. Here, the sampling, of course, should be random. When I say sampling here, then I mean the product. I'm not talking about the pharmacy, I'm talking about the product. Only in this paragraph. Lately, when I say sampling, then I mean the pharmacies. And there is a subset of sampling which is used to select the medications. I'm going to talk about that later. So the outcome here after the quality testing is the whole batch is either accepted or rejected. There should be a null hypothesis, and usually for the quality testing, we tend to be negative in our assumption. For example, we say the rejected units proportion is more than X% of the lot. Like we are expecting to find more than, for example, 10% of bad products out of this lot. And the formula of LQS then will tell me about the sample size and the decision rule. I'm going to mention this. But this is, these calculations are based on the definition of the upper and lower thresholds that classify good or bad lots or good or bad products. So for example, the manufacturer here can tolerate like let's say 20% of bad products. He has no problem with that. But more than that will make a trouble in marketing. So it's better not to send the product to the market. And the sample size and decision rule also depends on the probability of errors that here the investigator is willing to tolerate. I'm going to mention this in the second slide. But before that, I wanted again to talk briefly about this sampling method. This sampling method was proposed as an economic first step for monitoring outlets selling poor quality medicine. This was suggested by Professor Newton, as we see here. The bottom line is the reference. This sampling method requires smaller sample sizes than the simple random sampling or stratified random sampling method. Really small sample size. It is less costly, requires less time, less labor, less resources in general. But here, when we apply LQS here in sampling for the quality of medicine in the market, the lot here is not the product, it's not the medicine. The lot here is the number of pharmacies, or we call them the units here, with common characteristics, like they are within the same geographic area, or they all belong to private or to government sector, whatever my classification is. So the lot here is the pharmacies, it's not a batch of products. This method actually classifies the prevalence of bad outlets, those outlets or pharmacies that sell low quality or counterfeit medicines. They just classify the prevalence. It does not tell me exactly what is the prevalence. It cannot tell me that 15% of the pharmacies in this geographic area are selling counterfeit medicines, but it will tell me that the prevalence is more than 10% or 20%. This is based on the definition of the upper and lower thresholds and the errors, the type 1 and type 2 errors that we define. As I mentioned, this calculation method will give me the sample size and the decision rule. The decision rule here is the number of outlets or pharmacies that sell poor quality medicines. I would refer to them as the bad outlets that need to be found before an area is considered unacceptable. For example, if the decision rule is 5, then finding 6 pharmacies, which is more than 5, that sell low quality medicine will allow me to classify the whole batch of pharmacies in this region as unacceptable. There is one advantage here in sampling. It is actually related to the decision rule. I can stop sampling whenever I exceed this decision value. I can stop sampling. Let's say the formula calculated 40 pharmacies for me, and the decision rule is 3. Immediately when I reach 4 pharmacies that sell low quality medicines, I can stop sampling. Let's say I just finished 20 pharmacies only, but I found 4 of them selling counterfeit medicine. I can stop. No need to continue. This will save me money, time, and resources. So this is a very good benefit of this sampling method. This method is also useful for following up with these pharmacies. Like we can use it semi-annually, quarterly. It depends on what, and we can just monitor the changes in the market in a certain area. I have here from the WHO report. Here we can see in the bottom line down the recommendations on the content of survey protocols for surveys of the quality. This was a working document published in July 2014. It says random sampling LQS is probably the most efficient and accurate initial sampling procedure. And there was only one published LQS medicine quality survey here. I was really honored to be the primary investigator in this research, which was conducted in Riyadh, the capital of Saudi Arabia. So WHO referred to my paper. Another quote from another report by the WHO, which is the technical report series number 996, the expert committee on specifications for pharmaceutical preparations, the 50th report published in 2016. It says an alternative approach to formal random sampling that is simpler and less expensive and needs smaller sample sizes is LQAS. This technique can be used to determine whether the prevalence of outlets selling poor quality medicines exceeds a certain threshold. And again, they referred to one paper here. Says the examples of this approach are described in several publications. By this time, some other researchers have conducted other quality surveys. But again, one of my papers here was used as a reference by this WHO report, which is the quality of medicines in community pharmacies in Riyadh, Saudi Arabia, a lot quality assurance sampling based survey. There are several formulae that can be used with the LQS, but the famous ones are the binomial formula and the hypergeometric formula. I'm not going to mention those details here. Maybe they require a separate workshop actually. There is also a country scale LQS formula that can be used for surveys for the quality of medicine in a whole country. But usually these two formulae are used. What are the differences between them? The binomial formula is preferred if the population size is either unknown or very large. Let's say you have a geographic region, you don't know the number of pharmacies in this region. It's unknown to you, or let's say it's very large, it's in thousands. So in this case, it's better to use the binomial formula. While the hypergeometric model is preferred to be used if you know the population size, and if it's relatively small, we are talking about, let's say, hundreds maybe. So it's good to use it. I'm going to give an example about the null hypothesis here. We can say that we expect to see more than 20% of pharmacies in area X that sell low quality medicine. And another example here for the thresholds of acceptability of the outlets that sell poor quality medicines, I have to mention here that usually we don't like to find any pharmacy that sell poor quality medicine. So there should always be zero tolerance. But in fact, even in developed countries, even in the United States, in Europe, everywhere, counterfeit medicines were discovered even in the supply chain in these countries. So we always expect to see a certain percentage of outlets or pharmacies that sell low quality medicine. Here, in this example, I have used 20% for the upper threshold. So it means that it's not accepted for more than 20% of outlets to have low quality medicines. And the lower threshold was set to 5%. It means that it is acceptable. Acceptable, please differentiate between accepted and acceptable. So it is acceptable for less than or equal to 5% of outlets or pharmacies to have low quality medicines. Actually, when you define these upper thresholds. It's not arbitrary. It's based on other researches in other countries worldwide. And again, the reports of the authorities in the country or the city you are conducting the survey in. An example of the acceptable probabilities of error, we have the type 1 error, or we call it the alpha error. For the purpose of this kind of surveys, we like to call it the consumer risk. And actually in industry, they call it the consumer risk. It's the risk that is, again, acceptable for the customers when they buy a product to find it bad or broken or whatever. So an example here for the consumer risk is 5%. So it is the risk of accepting a bad lot or a bad batch. It should be less than or equal to 5%. Or we can say, on the other hand, statistically, that we are more than or equal to 95% confident that an accepted lot is a good lot. When you use 5%, it means a bad lot. 95% is a good lot. And we can calculate the confidence this way. The type 2 error, which is called also the provider risk here, is the risk that the provider, the seller, can tolerate. Usually it's set to be more than the consumer risk here. So I use here 10%. So it means the risk, it is the risk of not accepting a good lot. Let's say by chance, I rejected a lot by chance, while it is good. But I failed to accept it based on my quality testing, my sampling. So the risk here is set to 10%, less than or equal to 10%. So on the other hand, we can say that we are more than or equal to 9%, 90% confident that a rejected lot is bad. And again, we can calculate the confidence percentage. This is an example of the LQS calculator. You can find several online calculators online, like the Brixton Health calculator here for the hyper LQS, the hypergeometric model of LQS. What you need to do is you have to enter the population size, let's say 900 pharmacies in this city or the geographic area. The upper threshold is 20%, the lower threshold is 5%. The alpha error or the consumer risk here is 5%. The beta error or the provider risk is 10%. And automatically it will calculate the sample size. So I have to select randomly 36 pharmacies from out of these 900. You can see how small is this sample size here. The decision rule is 3. It means finding more than 3 bad pharmacies will make us reject the whole lot. So we can say the whole city or the whole region is rejected. It means that the counterfeit medicines or low quality medicines are prevalent, are more than, let's say, 20% based on our null hypothesis. So we can direct more resources to a certain area. And this calculator will show me the actual alpha and beta errors, which is almost 5% and 10% here. The decision rule, as I mentioned, it's the number of low quality medicines. So if the number of bad pharmacies exceeds the decision value here, so the lot will be classified as a high prevalence lot. So it means that more than 20% of outlets in this region sell low quality medicine, and the null hypothesis here is not rejected. Otherwise, if it's not more than 20%, immediately it will mean that the prevalence is below 5%. And actually, this formula just classifies the prevalence as either more than 20% or less than or equal to 5%. It does not classify in between. This is called the gray area, where you can call it the pool of errors here. So it just classifies as either done more than the upper threshold or less than or equal to the lower threshold. So it just classifies. It does not tell me the exact prevalence. This is one of the, maybe we can call it a disadvantage of LQS. But compared with the other advantages, it's almost nothing. I'll give you an example, a true example of LQS-based study. It's this paper. You can find it here in the bottom of the slide. One of my papers that were referenced by WHO. My objective was to classify private pharmacies in Riyadh, the capital of Saudi Arabia, in terms of the quality of medicines sold by them using LQS technique with a predefined threshold. My null hypothesis here was that more than 20% of private pharmacies, usually in Saudi Arabia, we have one kind of pharmacies for the public. We call it community pharmacies or private pharmacies. We don't have drugstores. We just have pharmacies that sell prescription, non-prescription, and some other health-related products. And we call them the private pharmacies because they are not belong to the government. So my null hypothesis that I was expecting to see more than 20% of these pharmacies in Riyadh, either in a chain or independent category. I classified the pharmacies into chain pharmacies and independent pharmacies. A chain, it means that we have 3 or more pharmacies that belong to the same owner, and independents mean 3 or less. So my assumption was or my null hypothesis was to see more than 20% of bad pharmacies in Riyadh. The sampling actually was conducted during 2010. The pharmacies were divided into chain and independent. The upper and lower thresholds were set as 20% and 5%, as I have mentioned in an earlier slide. The consumer and provider risks, or alpha and beta error, again, 5% and 10%. The sample size was 36, and the selection was truly random. I'm going to show you that later. The decision value was 3. So the second issue here was what kind of medication to collect from these pharmacies. There was actually a personal contact with the Saudi FDA and the Saudi Ministry of Health, because by that time there was no studies on the quality of medicines in Saudi Arabia. So there were more than one choice for me. I preferred just to collect amoxicillin samples. Amoxicillin without clavulanic, just amoxicillin tablets and capsules only, as quality indicator. So if I find low quality amoxicillin product in a pharmacy, then I may expect to see other medicines also, which are counterfeit or low quality. Amoxicillin is widely used. It is widely counterfeit also. On the other hand, it's heat sensitive. So even during bad storage or transportation conditions, the beta-lactam ring may break down, and we can lose the active ingredient. Actually, if it's stored in temperature higher than 30 degrees Celsius, within 1 or 2 weeks, more than 30% of amoxicillin will be destroyed. So amoxicillin capsules or tablets were purchased from each pharmacy, and actually I'm the mysterious shopper or the simulated client myself. By that time, it was allowed not allowed actually, but these private pharmacies were really selling antibiotics without prescriptions. These days, it's really not allowed, and they are being monitored by the authorities for this kind of behavior. And I've checked also for the authenticity. I contacted the manufacturers to check whether these products are authentic or not. Then the samples were analyzed for drug content and content uniformity, but not in Saudi Arabia. By that time, I was in Japan. I was doing my PhD in Kanazawa University in Japan. So I have to send the samples by DHL under very strict shipping conditions. So they have arrived cold and in special containers. Distribution map of the pharmacies. This is Riyadh, and you can see that really samples were randomly collected. These red spots represent the pharmacies I collected from. So it is true random sampling. Unfortunately, I have found substandard, not counterfeit, but substandard and some degraded samples of amoxicillin in 9 pharmacies, 4 chain pharmacies and 5 independent. For the degraded medicine, actually, the amoxicillin, I mean, it was a manufacturing defect. I'm not going to mention actually the manufacturer because it is confidential. I can declare it only to the authorities, and I have did that already. Other substandard samples were actually authentic, but it seems to be that they were stored in very harsh conditions. I mean, heat, high temperature, and maybe humidity. Unfortunately, also during sampling, I could not stop sampling because I don't know, I just collected samples. If I was having the X-ray machine or the infrared, whatever, so I can check on site. Once I reach the decision rule, I can stop. So there's no need to continue to waste money and time. But I couldn't do that because I had to collect the samples and take them with me to Japan for analysis. So both lots here were rejected, the chain and independent pharmacies, because I have exceeded the decision rule here, 4 and 5. So they were rejected as unacceptable, which may indicate that consumers in Riyadh may be at high risk of purchasing substandard medicines at community pharmacies. Fortunately, this is the final slide. In conclusion, the quality of medicines sold in Riyadh were found to be not meeting the acceptability criterion, which I set for the upper and lower threshold. It means that more than 20% of pharmacies in Riyadh at that time were selling low quality medicine, not counterfeit, but low quality or degraded maybe. So I have recommended some interventions by the authorities, and actually they responded, and I recommended that there should be a follow-up studies, but until the time, this time actually there's no follow-up study was done. Maybe it's a good idea for me to do it again. So LQAS again proved to be practical, economical, statistically valid, reproducible sampling method for the quality surveys. Thank you so much. I hope that you find this presentation useful. Thank you again, the College of Pharmacy and Assiut University. Special thanks to the Dean, Dr. Mahmoud Shiha, for the invitation. Thank you and have a good day.
الرحمن الرحيم. يسعدني أني أكون من المدعوين على مؤتمر الكلية الدولي ال 12. وسعيد أكتر إن أنا في بيتي الأصلي كلية صيدلة جامعة أسيوط، كليتي الأم. وتحياتي وتقديري للسيد الأستاذ الدكتور طارق جمال رئيس جامعة أسيوط، السيد الأستاذ الدكتور أحمد المنشاوي نائب رئيس الجامعة لشؤون الدراسات العليا والبحوث، السيد الأستاذ الدكتور محمود شيح عميد الكلية، الأستاذة الدكتورة جهان فتيح وكيل الكلية لشؤون الدراسات العليا، السادة الحضور، أساتذة الكرام وزملائي الأعزاء. النهاردة هنتكلم على الموضوع اللي هو يعني سلايد فور بروب، ولكن عشان نفهم الجيرني اوف ذيس ريسيرش او الهيستوري اوف ذيس، هنتكلم على design and synthesis of novel triazole derivatives as small molecules cytohistine inhibitor culture drug candidates. Overview of the presentation includes cytohistine work design, synthesis of new derivatives of CSNH3, study of SAR of these compounds, multigram scaling of CSNH3, and biological results and applications of this project. First of all, G protein are an important class of regulatory switch in all living systems. They are activated by guanine nucleotide exchange factor, gives protein, which facilitates the exchange of GDP for GTP. This activity makes GIFs attractive for modulating disease. A relevant G protein controlled signaling network. GIF inhibitors are therefore of interest as tools for elucidating the function of these proteins and their therapeutic intervention. If we look to G protein, we have two classes of ARF GIFs are known and can be distinguished by their size. Cytohistine belong to small ARF gives proteins around 47 kilodalton. The large one ARF gives protein, which also contains CES7 domain, have a mass of about 200 kilodalton. Unlike the large ARF proteins, or ARF gives protein, a small cysteine H3 responsive cytohistines are insensitive to the fungal metabolites prefilled. Discuss this reagent later. Cytohistines are class of PFA resistant small GIFs for ADP ribulization factor اللي هم الARFs protein, which regulate cycloskeletal organization, integrin activation or integrin signaling. As you see, the composition or domain composition of cytohistine 1 and their human homologs consists of CC coils containing domain protein-protein interaction, CES7 domain bearing GIF activity, PH plexigen homology domain mediating recruitment of plasma membrane by binding of photo inositides. The only one small GIF CES7 domain inhibitor is the RNA aptamer. Some people look to the title. Some people look to the title when we say small molecules, and if we are looking to our molecules, it's not actually small enough. But if you compare our compounds to the only reported aptamer M69, which represses the green nucleotide exchange in vitro and reduce T cell adhesion repression, you can look to it. It's aptamer. It's actually huge, large compound. That's why we assigned our compound as small molecules cytohistine inhibitors. The small gives inhibitor refill. We just announced it, but it is not specific, and also small ARF proteins are resistant to this agent. Prefilled E was initially isolated as antiviral, is isolated as antiviral, but it is now primarily used in biological research, the protein transport. It's a natural product or fungal metabolite. The history of cysteine H3 or the application of cysteine H3 started by publication Shell found book in Nature in 2006. Inhibition of cytohistin by cysteine H3 leads to hepatic insulin resistance. This the small compound triazole derivative cysteine H3. The application of cysteine H3 in human liver cell showed that insulin receptor complex associated with cytohistins are required for insulin signaling, and this actually it is the attractive starting point with cysteine H3 derivatives journey in the research. Cysteine H3 treated mice show increased expression of gluconeogenic genes, reduced expression of glycolytic genes, reduced fatty acid and ketone body metabolism genes of the liver, reduced liver glycogen store, and compensatory increase in plasma insulin. Thus, cytohistin inhibition results in hepatic insulin resistance, because resistance is among of the earliest pathological change in type 2 diabetes. This result shows the potential chemical biology for dissecting the molecular pathogenesis of this disease. In the beginning, we tried to make new derivatives of cysteine H3 as cytohistin inhibitors, and more or less the way was blind in the synthetic pathway because we have no any idea about the of cysteine H3. So we began by drug design or postulated drug design for synthesis of these new derivatives. Actually, the first part we did take in mind is the phenyl group is essential or could be replaced by another heterocyclic ring. The second is the sulfur essential to be exocyclic or could be included in the hetero ring? Is it essential at all? If we remove the sulfur, it's essential for the activity. What was the second question? The third question is the piperonyl moiety is essential or could be replaced by electron withdrawing group in the phenyl ring? The fourth and last question for us, is it favorable with the new change or should be replaced by ethyl or another alkyl group? I mean the methoxy group attached to triazole moiety. So we had four questions. We must answer it, or we must dissolve it, or we must investigate it to synthesize new derivative more potent and more selective than cysteine H3 itself. Since of new derivative of cysteine H3 study and SAR of this compound, cysteine H3 actually is an exciting compound as cytohistin inhibitors and insulin resistance induced to study the SAR and try to find better candidates of cysteine H3 derivatives. According to our information or to the information actually we did take or we taken from computer modeling drug design done with Professor Jürgen Piorat. All the new derivatives have log P less than the cysteine H3 itself, and then increase the solubility of these expected new derivatives, and this is a big advantage for any biological active compound. The chemistry pathway is too long, so I will contract the pathway. Started from acid chloride. In multi-step reactions, we got the trinitro triazole nitro compound, and we reduced it to amino group and then coupled the final thiomoiety to this to prepare our derivative. You see lots of derivatives of CSNH3. Derivative is synthesized through protein chemistry, and some derivatives were terrible in synthesis, but we did. Compound or derivatives was synthesized in this project, as you see, and we tried to change replace the phenyl moiety or delete the sulfur atom or include the sulfur atom in heterocyclic rings that find thiophenyl derivative, or replace the low moiety by electron withdrawing group, and to study or to compare which is better, electron withdrawing group. We all used electron donating group for these derivatives, such as H107. We introduced to the phenyl group tertiary butyl moiety instead of electron withdrawing group, to trying, or in our trials, to make SAR or structure activity relationship of this derivative at the end. We tried to oxidize the sulfur to sulfonyl moiety to study the hydrogen bonds probability to the two oxygen of sulfonyl moiety. We also faced many problems of AH228 28 as a negative control. It was crazy, terrible, but we did. After many trials, on as illustrated in this scheme, to introduce this moiety and replaced the methoxy group by isopropyl moiety, and this compound used as active control because it's not active actually in as a cytosine inhibitor, but after many trials we got this compound, as you see, like a foam. It was funny. After 1 month's work, I did it. We are not going now to the details of chemistry obstacles we faced in these compounds. After that, we faced another problem, gram scaling synthesis of CSNH3 and their derivatives, because we needed it in vivo investigation as potential insulin resistance inducing agents, and the mice enjoy it. In gram scaling, we optimized the procedure fastly, just change the method of reduction from chemical to catalytic reduction using autoclave, as you see, and the yield is improved from 34 around more or less to 64 or 70% after reduction using autoclave, and at 24.7 gram of CSNH as standard compound for cytohistine inhibitor in vivo experiments. The biological results of these new derivatives: sulfur atom is essential for activity. Terminal thiophenyl group is critical for activity. Deletion of the thiophenyl residue results in dramatic loss of the activity, such as we replaced the thiophenyl by COSH3, led to dramatic loss of the activity. Conversion of sulfonyl group to sulfonyl didn't enhance activity, but didn't abolish it. In conclusion, we concluded the inclusion of sulfur in a heterocyclic ring didn't affect the activity of some compounds, whatever increase or decrease. The alkoxy substituent of 3 position of triazole ring is essential, and the best was methyl group, and in some cases ethyl moiety. Replacement of phenyl ring heterocyclic ring is possible, and sometimes increased the activity such as pyridine, thiophene, pyrimidine, thione, and so on. Presence of electron attracting group on the phenyl ring found to be essential. But our surprise or big surprise, the best compound was found is AH107, in which we introduced alkyl tertiary alkyl group as electron donating group. Actually, in the beginning, we introduced this moiety for comparison of the activity between electron donating and electron attracting group, but this compound was 3-7 times more potent than cysteine H3, more potent than cysteine H3. This, in brief, the QSAR of this derivative. From these biological or from these derivatives, we applied the biological results of cysteine H3 new derivatives in the design and sense and application of bifunctional cysteine H3 photoaffinity probe for specific photoaffinity labeling of cytohistine cysteine 7 domain. Cysteine H3 derivatives target the 7 domain of cytohistine, also known as alpha-nucleotide binding site opener or ARNO, and cytohistine 3, and inhibit their guanine nucleotide exchange activity. Its application in human liver cell flies and mice led to impaired insulin signaling, and in each case. And our main target in this, the second project, or the first biological applicable project from these derivatives, is the bifunctional cysteine H3 photoaffinity. So we synthesized the cysteine H3 as a standard compound to compare other derivative with this cysteine H3, but we are looking for a photolabeling moiety. So we synthesized XH1009, as we discussed before, replaced the phenyl thio moiety by acetyl group, and this compound actually not more or not much active if compared to cysteine H3. We prepared it for comparison actually. After synthesis of the negative control, we can say, we synthesized cysteine PP, in which we introduced the benzophenone moiety, and then we synthesized biocysteine H3 and cysteine PP. In biocysteine H3, we introduced the biotin biotag as photolabeling moiety, and finally we got biocysteine H3, which contain the biotag moiety, as you see, as labeling group, and then we introduced this biotin tag labeling group to the cysteine PP, and we got finally the biocysteine PP, which bifunctionalized it as photo-labeling reagent, which we apply it as bifunctional for cytohistine cysteine 7 domain. The history of the chemistry is too long, but I try to collapse it because of time. So after we got these 3 derivatives, after much work in chemistry, the biological application inhibition by cysteine H3, cysteine PP, and XH1008 of guanine as illustrated. We see the inhibition by cysteine H3, our main compound, our target compound, and this our new synthesized compounds by functionalized photo-labeling compound with cysteine PP, and this the inhibition using XH1008, the inhibition is not so much, or we can say it is not active. So after that, we applied our compound biocystin PP to the RNO-CIS7 domain, specifically after radiation. As you see, D biotin and his sex, and his is the relative optical intensity of RNO-CIS7 domain. As you see, the labeling of RNO-CIS7 domain with biocystin PP was increasing radiation time. If we increase the radiation time, the photo labeling is increased, and we fixed the concentration in all cases 2.5, 2.5. So the radiation, or the optimum radiation time for labeling, is 3 minutes. After that, the amount of labeled protein increased steadily as a function of time. After irradiation for 3 minutes, the amount of labeled protein was 30 times greater than that observed for non-irradiated background, as you see. After that, the photoaffinity labeling of RNO-CIS7 domain with biocystin PP as a bifunctionalized labeling agent, increasing with the concentration. We studied the optimum time for irradiation, and we studied the effect of concentration of our reagent. As you see in biocystin, our biocystin PP, our labeling agent, starting from 0 till we reach to 5 micromole, the affinity, how the labeled protein of RNO-CIS7 domain to our agent on biocystin H3 2.5, and this, its important point, was subjected to the same condition. The signal intensity was similar to that of background, which shows that the labeling depends on the presence of the photophore. Means our photophore here is the highest one in optical activity of RNO-CIS7 domain. We just added the biocystin H3, the affinity, or the labeled protein is dropped, and this is a pure evidence that the photo labeling of RNO-CIS7 domain protein is labeled by our reagent biocystin PP. And then we applied the biocystin PP in presence of cystin H3 and in presence of H1009, as you see here, S domain and D bio. And we used 2.5 in all cases of biocystin, then H3 2.5, and the photolytic is more or less disappeared. Competition experiment was also performed to verify that the binding mode of biocystin PP was similar to that of cystin H3. We want to prove that the affinity is in a competition study or competition affinity towards the RNO-CIS7 domain, and this is proved by application of our functionalized reagent with our cystin H3. As you see, the labeling is disappeared. In the presence of equimolar amount of cystin H3, the amount of labeled protein, as I said, decreased significantly. In contrast, XH1009, its concentration didn't affect the affinity of photolabeling, because we discussed before that XH1009 has no any affinity towards ARF or GIF protein, or specifically RNO-CIS7 domain. To verify that biocystin could be used as a bifunctional probe, or we examined the our labeling specificity by using a rank of ARF-GIF CIS7 domain, GIFs without CIS7 domain, and small GTPase enzyme with a large variety of ARF protein, including ARF6 and RAS, and for example, CIS7 domain and the CREB1 CIS7 domain. Finally, a photophore probe for cytohistine was designed, synthesized, and applied by integrating the benzophenone group into cystin H3, such a way that its inhibitory activity was maintained. Further functionalization with detectable biotin tag led to biocystin PP based our activity based protein profiling, APP, reagent for cytokine. The application of this probe to a variety of GIFs protein and GTPase provided a cogent evidence for the specific binding of cysteine H3 to the cis7 domain of the membrane of cytohistine family. Reaction conditions could be defined now that may enable the proteome-wide affinity-based profiling of cytohistine complex on a whole cells or in even organisms. And this above mentioned work is published in Adjuvant Chimie. The last application for this project, slight ROBO4 signaling promote vascular stability by blocking ARF6 activity, and this was published in Nature Cell Biology 2009. Slight roundbot ROBO signaling has a well understood role of axon guidance, unlike the nervous system. However, slight dependent activation of an endothelial specific ROBO4 doesn't initiate guidance program. Instead, ROBO4 maintains the barrier function of the mature vascular network by inhibiting the new vascular tuft formation and endothelial hyperpermeability induced by pro-angiogenic factors. In the study, we used cell biology, organic chemistry, and biochemical techniques to elucidate the molecular mechanism underlying the maintenance of vascular stability by ROBO4. Here, we demonstrate that ROBO4 mediate slide 2 dependent suppression of cellular protrusive activity through direct interaction with the intracellular adaptor protein Paxline and its paralog HEC5. Cumulative, these data demonstrate the importance of Paxline and GTI and GIT1 in mediating slide 2 ROBO4 dependence, inhibition of endothelial cell protrusive activity, and suggests the blocking activation of ARF6 protein is a potential therapy for human disease characterized by pathologic angiogenesis and the vascular leak. In very brief summary, as you see, we have many pathway in activation of or block of ARF, and the blocking of ARF led to blocking of RAC protein, led to migration on angiogenesis and vascular leak. In a simple way, there was some suggestion or some possibility that slide 2 for complex with Paxline RNA with each other block ARF protein and RAC led to migration of angiogenesis and vascular leak. But was no evidence about this matter. So people or our partner in San Diego and Florida seeks a reagent proof that the block of ARF6 and RAC led to migration of angiogenesis and vascular leak. So VEGFR, vascular epithelial growth factor receptor, and integrin signaling remote sequential activation of ARF6 and RAC, leading to cellular protrusive activity, concomitant initiation by ROBO4 signaling by slide to stimulate the recruitment of Paxline ARF cap complex to cytoplasmic tail of the receptor, which cause the local inactivation of ARF6 and RAC, thereby blocking the protrust activity necessary for angiogenesis and vascular leak. And after the application of cysteine H3 in a parallel way in Arno, we, as you see here, VEGF 165 and cysteine H3 with each other in common manner, and here the GTP R6 in this slide A and B, lysate from endothelial cell pre-treated with cysteine H3 or DEMSU, then stimulated with VEGF 105, were precipitated with GSD, GGA3, and immune with ARF6 antibodies. In B diagram, endothelial cells pre-treated with cysteine 3 or DEMSU were subjected to migration assay using VEGF 165. And also, we confirmed this in another way, quantitation or quantification of pathologic angiogenesis observed in 15 animals. As you see, laser-induced coronal neurovascularization, here in the presence of CSNH3, here in the presence of Dempsey control, here also VEGF induced retinal permeability, here in the presence of CSNH3, and here is with Dempsey as control. And at the end, consistent with this in vitro study, inhibition of ARF6 activity, inhibition of ARF6 activity in vivo phenocopy's ROBO4 activation by reducing pathologic angiogenesis in choroidal and retinal vascular disease and VEGF 165 vascular and cellular growth factor induced retinal hypermeability. And these data reveal that slide 2 ROBO4 Paxillin GITI network inhibits the cellular protrusive activity underlying new vascularization and vascular leak, and identify a new therapeutic target for ameliorating disease involving the vascular system. For the first time, proved that slide 2 ROBO4 Paxillin play an important role in this vascular leak or neurovascularization of the retinal membrane. Paper published from this work, specific photoaffinity labeling cytosensitive domain was bifunctional CSNH3 photoaffinity probe angiopantochimie. Slides 2 ROBO4 signaling promote vascular stability by blocking ARF6 protein. And this blocking discussed, it is proved the rule of ROBO4 by parallel block using CSNH3 as a specific blocking agent for ARNO is published in Nature Cell Biology. Design and sense effect of CSNH3 related cytosine analogs of ARF activation in insulin-dependent transcription still in study and in progress for general medicinal chemistry for submission in journal medicinal chemistry, and I think more or less around finished. Design and synthesis of new CSNH3 as potential cytoresin antagonist, Indian Journal of Pharmaceutical Science, 2014. Outlook or our current project, xanthine derivative as PepsiK inhibitors as potential anti-cancer, and we published from xanthine derivative as potential anti-cancer last year 2 papers in bioorganic chemistry, and this year 2 papers in bioorganic chemistry, and we have now 2 papers in progress, one for European Journal of Medicinal Chemistry and one for bioorganic. As you see, caffeine increase alert and mood, and I am addict for caffeine. Thiophyllin increase the fresh air as bronchodilator. So our current projects with my group now is concentrated in xanthine derivative or focused in xanthine derivative. Another project with Michel Famulek in Bonn, Germany, using of trifurcated as DNA intercalator using organic chemistry and bioorganic chemistry and biological chemistry and DNA blocking or basic DNA trifurcated or DNA intercalator. I think it will take much time because it's complicated. As you see, a major and minor groove, and our idea in our basic site in minor or major groove. To thank you all for your good listening. Great thanks for Michel Famulok, Alexander Heckel, Christina Herman, and my lovely group, Jeff, postdoctors, and Jiha, in addition to Ackerman from Switzerland, and this also my groups, and to my supervisor. And till now, we are incorporating in many projects, Chris Moller in Bonn, Stephanie in Company, and Britta Schumacher, the head unit community of Roche Company, and Juliet, this of the chess group of Michel Famulok and this group, I really like it because we are in cooperation till now. Thank you very much. And again, I'm very happy and thank you for my invitation to this respective conference and to my lovely mother faculty. Wassalamu alaikum wa rahmatullahi wa barakatuh.
First, I want to introduce myself. Awal Abu Radwan Salama, Professor of Pharmaceutical Organic Chemistry, a PhD Computer Aided Drug Design from Kitasato University, Tokyo, Japan, 2002. Structure and ligand-based database search to discover antibacterial inhibitors of Staphylococcus aureus histidine kinase enzyme. Drug discovery is a long, expensive, and risky process. It needs about 15 years and between $1 and $2 billion. It has high attrition rate. For 10,000 molecules tested, 10 molecules passed to clinical trials. Only one compound passed to the market as a drug. 70% of the costs are wasted on failure. The drug discovery process is a very wasteful game. Based on the Pfizer pharmaceutical company, about 100 discovery approaches tested millions of compounds. Only 1 to 2 compounds passed to the market. The drug discovery process, the steps start from identification of the disease, identification of the target macromolecule, identity of lead molecule, optimization of lead molecule, then preclinical trial test, then clinical trials, and the last step is approval, FDA approval of the drug. This, as you see, it takes from 11 to 15 years and cost millions of dollars. The database searching is target of the pharmaceutical companies to save money and time. There are 2 types of database searching, either screening in laboratory or virtual screening. The screening in laboratory, although it is optimized process, but it is still very expensive and time consuming. So the pharmaceutical company have been directed to the virtual screening of the databases to search for leads as pre-stage for experimental testing. What is the virtual screening process? The virtual screening process is the use of high performance computing to analyze large databases of chemical compounds in order to identify possible drug candidates. Drug discovery information piled up over the past years are stored in and categorized into 2 major categories, chemoinformatics and bioinformatics. What about the size of database? Databases are of huge size data. It contains millions of entries for the chemoinformatics and several thousand of entries for bioinformatics. Fortunately, collaborative computing applied in computational chemistry allows users to share documents stored in chemoinformatics and bioinformatics databases. Today, the virtual screening is a very common approach to identify hit compounds from large libraries of molecules in the drug discovery process. It can be classified into ligand-based database search, structure-based, then de novo structure-based. In the ligand-based database search, it depends on the use of either one-dimensional, two-dimensional, or three-dimensional structure of the ligands. The structure-based database search depends on the use of the target macromolecule as a query for database search and using available commercially or academic available docking programs as a representative programs, Autodoc program, Doc program, FlexX, Glide, Gold, Surflex. Each program has some strengths and other and weakness. Advantage and disadvantage of virtual screening. The advantage: no need to synthesize new chemicals, fast, cheap. Disadvantage is false positive rate, limited chemical space, and possibility to discriminate the intrinsic activity, necessity to confirm the results by experimental assay. Our case study is virtual database screening to discover histidine kinase inhibitors as potential therapies of Staph aureus infections. Our resource in this study is information about the disease. Second is the target macromolecule. The third is available active drugs against the disease. First, our information about the disease is the pathogenic Staph aureus can cause a range of illness from minor skin infections to life-threatening diseases. Pathogenic strains often promote infections by producing virulence factors such as potent protein toxins. About our information about drugs, there are few active compounds that control the expression of virulence factors required for infectivity of Staph aureus. We have limit chemoinformatics. Our information about the target is a two-component signaling system mediated by histidine kinase is the target macromolecule in our case. The virulence, briefly, the virulence of pathogenic bacteria is an adaptive genetic response. That means pathogenic microbes must be able to sense when it is in an invasion position, then reacts as a result of gene coding, producing a final pathogenic response. This adaptive response is a vital for survival in harsh environments. This response is mediated through two-component system that is ubiquitous in bacteria, absent in humans. Only amino acid sequence is available for Staph aureus, two-component system. The three-dimensional structure is unpublished yet. The Staph aureus two-component system comprises membrane-bound sensor histidine kinase walk part and response regulator protein. Walk a membrane-linked protein or membrane-linked histidine kinase consists of catalytic or ATP binding domain and dimerization domain. And response regulator protein composed of response regulator domain and DNA binding domain. The biochemical pathway of this molecular target, first, interacting with external stimulus with the extracellular signaling monomer of histidine kinase results in dimerization and ATP and activation. Then ATP binds to the walk domain. Then transfer of a phosphoryl group from the ATP to histidine on the dimerization domain and the phosphotransfer domain. The phosphoryl group transfer from the histidine to the aspartic acid conserved on the response regulator protein. Phosphorylation of the response regulator protein results in its activation and subsequently triggers it to bind to DNA and stimulates the gene transcription, finally producing the response in the form of toxins and producing the infection. From this biochemical pathway, we can see that ATP binding to WOC is crucial in this process. So inhibiting ATP binding to WOC will inhibit the bacterial response and inhibit bacterial infection. Thus, bacterial activity can be achieved by inhibiting microbial histidine kinase WOC using ATP competitors. Few inhibitors of Staph aureus histidine two-component system, and only there is amino acid sequence available for the target macromolecule. That means the three-dimensional structure of the target macromolecule doesn't publish yet. Now we have from the bioinformations only the sequence of the target, and from the hemo information we have a few number of compounds, active compounds. Waldmycin is active compounds, and the waldmycin ester analog is almost inactive. How can we use this data to search the data as a query for searching the database? First, the amino acid sequence is used for building the three-dimensional structure of the binding cavity, so using complex modeling. Can be used, the modeled binding cavity can be used for database searching. First, let's try to look active and reactive compounds into the binding site. Unfortunately, the resulting solutions of the docking process revealed that the almost similar binding orientation for active and inactive compounds, and almost equal docking score. For this reason, using this docking process for database search in our directory couldn't discriminate between active and almost inactive compounds. For this reason, molecular dynamics simulation has been done in order to discriminate between the active and inactive compound through the highly sophisticated calculation process. Then that could discriminate between the active and inactive compounds and have different orientations and different free binding energy. For this reason, combination of the docking and molecular dynamics simulation and the other information of the docking obtained are used for database searching process. This is sketch representation of the overall workflow. The first step is first we have two-dimensional structure or amino acid sequence of the target molecule. Sequence alignment and homology modeling. The obtained three-dimensional structure from homology modeling used as input for nuclear dynamic simulation in order to optimization of the nuclear structure. The optimized binding cavity was used for docking of the active compound and inactive compound. Then molecular dynamic simulation, first step, molecular dynamic simulation to check the stability of the docked conformations. To check the stability of the docked conformation using molecular dynamic simulation, that the docked conformation either exit outside the binding site, that means false docking, or still in its binding site, that means stable docked conformation, or repositioned to another position or another binding site. Then molecular mechanics calculation has been made to calculate the binding free energy to rank the active ligands. Then fifth step is database search using this combination of the protocols. In homology modeling, briefly, sequence database search module was utilized to get the X-ray structures similar to the target sequence and rank them based on similarity percent. The top-ranked PDB entry was selected and used for building the homology model. The homologue model was checked for missing atoms, residues, and was repaired. This homologue model was optimized using MD simulation process. This is a result of homology modeling process. This is an obtained homologue model of the histidine walk of Staph aureus. The nuclear dynamic simulation for the optimization of the homologue model structure using AMBER 18 program. The homologue model was optimized in an aqueous solvent box to simulate the real physiological environments. During the MD simulation, the stability of the structure was calculated by its deviance from the starting conformation in root mean square deviation terms. This is the Ramachandran plot of the homologue model and the MD simulation obtained structure in order to check the correct position of the side chain of the amino acid of the build molecule, of the build target macromolecule. See that Ramachandran plot is a good orientation of the side chain of the amino acids. The binding mood generated by molecular docking, as we said, according to the docking study, almost failed in assessing the ligand binding affinity that couldn't differentiate between active and inactive. Same with same binding orientations and the same docking score. Fortunately, considering both protein structure flexibility and protein ligand electrostatic interactions, molecular dynamic simulation were deemed necessary to justify the activity difference between valdomycin and its ester analog, and to refine the understanding of their binding modes obtained from the molecular docking. The result of molecular dynamic simulation was different for waldemicin and waldemicin ester. For waldemicin active compound in complex with histidine kinase O, both the waldemicin and the protein structures were equilibrated with an obvious root mean square deviation fluctuations through the molecular assimilation process. However, with waldemicin methyl ester analog, the weak active compound, and with a root mean square deviation fluctuation over the first 11 nanoseconds, then showed a gradual increase of the RMCD deviation fluctuation between 11 and 14 nanosecond. Then quick increased RMCD fluctuation between 14 and 14.5. After that, it converged until the end of the process. This what happened with waldemicin methyl ester analog. The large RMCD fluctuation suggests large conformational change and repositioning of the waldemicin ester analog inside the binding site, resulting in 4 conformations. Remarkably, trajectory analysis revealed that the ester group exited the binding site after 11 nanoseconds. This conformation instability during MD simulation is unsurprising, since weak binding affinity was reported for this binder. This is a video recording of the waldemicin ester behavior inside the binding site. In the first video, water molecules were removed for clarification, and lysine amino acids were sketched in lines. Immediate simulation of the duct complex. Unfortunately, the video didn't work, or doesn't work. I don't know why. Immediate simulation of the duct complexes. The final snapshots of the ligand protein complex in MD simulation. The ligands are represented as sticks. The V-shaped orientation is for waldemicin, the active compound, and the extended conformation is for the waldemicin methyl ester analog, the inactive or almost inactive compound. Also, molecular mechanics pairwise decomposition analysis of residue interaction energies of the high occupancy hydrogen bonding residue to the ligands. The residue that make hydrogen bonding or with high occupancy during the MD simulation was selected, and its participation in the free binding energy have been calculated. From the result, it appears that lysine 100 and valine 180 has a high participation in the free binding energy. Also, total binding free energy calculation using molecular mechanics Gibson method have been done, and it shows the preferred free binding energy for waldemicin over waldemicin methyl ester. This part of work has recently been published. This is the second part is going. in continuation of the previous discussed work, in this section, I am going to walk you through the next part of virtual database screening. Now we have both bound conformation of vancomycin and binding cavity of the modeled histidine kinase walk proteins. This information will be used to screen the database. In first step, ligand-based database search was done using the similarity with the active compound vancomycin drug. Then the obtained diversity subset library undergoes structure-based database search using docking molecules program. The obtained candidates from docking will be visually checked for binding orientation, hydrogen bonding with lysine 100 and valine 184, and the smaller number of compounds or obtained candidates will be used as input in molecular dynamic simulation and molecular mechanic GIPSA to calculate the free energy of binding and will rank it based on the free energy of binding and binding rotation, hydrogen bonding with lysine and valine, and the resulting candidates will be suggested for experimental or laboratory testing. These are the preliminary result of database search, but not final result because the work is ongoing. Show the vancomycin with some retrieved candidates from the database. Thank you.
السلام عليكم.
السلام ورحمة الله وبركاته.
السلام ورحمة الله وبركاته.
دكتور هاني، أنا بنشكر حضرتك على البرزنتيشن الهايلة دي، وكان في بس كام سؤال لو حضرتك تسمح بيهم.
اتفضل.
أول سؤال كان مبعوت لنا: why couldn't we stop the sampling after exceeding the decision rule?
Actually, in my study, I couldn't stop sampling just because I wasn't having the instruments for on-spot analysis. Like I mentioned in my presentation, if I would have the X-ray detector or the infrared detector, then it would be very easy for me, just when I reach the decision value, just to stop immediately. At that time, in 2010, when I conducted the study, actually, I wasn't having that instrument, so I have to send the samples to a special lab. Actually, I sent it to Japan, where I was doing my PhD that time. So this is why I couldn't stop sampling. Otherwise, it's very easy. You can stop, and you save time, you save money by that.
شكرا. طب السؤال الثاني حضرتك: factors should be considered when we aggregating results obtained from the surveys.
Sorry, can you say that again, please?
Factors should be considered when we aggregating results obtained from LQAS surveys. Factors.
Yeah, actually,
okay.
the factors that should be aggregated, considered.
Yeah. After aggregating results obtained from the survey.
Well, what I understood from this question is that you mean the reporting,
تمام.
the reporting of the results, you mean?
أيوة بالضبط.
Well, actually, it depends. Are you doing this for your own research or is it for the government? Of course, when you have the results, you should declare them to the authorities in your country, so they will be aware of the high-risk areas where counterfeit or low-quality medicines are available. This is what I understood from your question.
Yeah. Okay. السؤال الثالث. Can we use this system or this survey in heterogeneous community?
Yes, of course. It depends on how you divide this community. So you can apply LQS in just one community, whatever, homogeneous, heterogeneous, or you can divide the geographic area or the country into sectors, and you apply the LQS in each sector. You deal with each sector as a lot, a batch of pharmacies. So it's very easy to do that.
تمام. بنشكر حضرتك واسفه على اذا كان في غلط في التقديم.
Yeah, actually, you introduced me as the dean of the College of Pharmacy in Taiwan University. I'm the vice dean, and I'm an associate professor, not assistant.
استاذ مشارك. انا اسفه.
Thank you so much.
شكرا دكتور هاني. هو بس يعني يمكن حضرتك عارف في مصر هنا في الاسيستنت بروفيسور بنقول عليه ليكشرر او استاذ، فيعني يمكن ده مع الزميله العزيزه الدكتوره نهى ده الحدث يعني، لكن الالقاب محفوظه به وحضرتك نار على علم يعني. اهلا بحضرتك.
بارك الله فيكم، جزاكم الله خير، واجمل وافضل تحيه لكم من مدينه رسول الله عليه افضل الصلاه والسلام.
عليه الصلاه والسلام، وبلغوا منا السلام ولاهل المدينه الكرام جميعا يعني.
ان شاء الله.
شكرا. يعني يسعدني ارحب طبعا بابن الكليه والزميل العزيز الدكتور عواد عبده رضوان، وهو طبعا من البايونيرز في الكمبيوتر ايدد دراج ديزاين. يعني هو اول يعني بعثه طلعت كانت فول اووردد تو ستادي كمبيوتر ايدد دراج ديزاين من جامعه اسيوط. كمان يعني من الناس الدكتور علاء دخل متاخر فاحنا معلش سكيب دي. دكتور علاء هنرجع لك ثاني بقى. دكتور عواد برضو من الناس الاوائل في جمهوريه مصر العربيه حقيقي اللي اشتغلوا في المولكيولر ايدد دراج ديزاين، وهو طبعا حاليا في جامعه الملك سعود بالمملكه العربيه السعوديه. وبنرحب بيك مره ثانيه دكتور عواد في بيتك.
اهلا وسهلا دكتور طارق، الله يبارك فيك، وحشتنا. الله يبارك فيك. وعلى دكتوره ازي حضرتك يا دكتوره؟ الله يبارك فيك. ازي حضرتك يا دكتور؟ الله يبارك فيك.
دكتور عواد، يعني how the homology modeling is confident, how you did يعني get a confident or trusted model to be used for design of new molecule.
في الاول برحب بالساده السبيكر اخواتنا الدكتور علاء عرفات والدكتور هاني، وبرحب بحضرتك والدكتوره نهى. بالنسبه للهومولوجي موديلنج ده ساينس يا دكتور طارق اوريدي استابليشد، وده بيستخدم لما يكون انا عندي في بعض البروتينز زي بعض الانزيمز او الريسبتورز زي اللي دي بتبقى كابلد مع الممبرين، وبالتالي صعب يحصلوا عليها عن طريق الاكس راي، لانه عشان اول ما يقطعها من البروتين يقطع الريسبتور او يقطع الانزيم من البروتين اللي شابك عليه فبيفقد الشكل بتاعه، فبالتالي ما يقدرش يعمل له اكس راي كريستاليزيشن. واضح حضرتك؟ وبالتالي في الساينس طلع اللي هو الهومولوجي موديلنج ان ممكن يستخدم السيكونس 2 دايمنشن سيكونس. يعني انا عندي السيكونس بتاع الانزيم او بتاع الماكرو مولكيول عن طريق بيشوف شبيه ليه على موجود في الداتابيز اكس راي ستراكشر شبيه ليه بشرط التشابه ما بيقلش عن 30%، مع ان في ابحاث ناس نشرت هومولوجي موديلنج على تشابه 25 و20%. ابحاث اوريدي ريبورتد على تشوع 2020 وطلع موديل وزن 4 انجستروم ار ام سي دي ديفيشن من ده بيعمل بيلد على ياخد السيكونس وبيعمل بيلد زي ما يكون بتدي واحد قماشه وبتدي موديل تقول لي فصل لي على الموديل ده القماشه دي. واضح حضرتك؟
انا يعني كان سؤالي دكتور عواد تحديدا هاو اي تراست ذا كرييتد موديل في ان انا اعمل ديزاين للمولكيول. هل المولكيولز اللي حضرتك يعني جربتها عليه او عملت دوكينج بيها عليه، هل يعني في حاجه اتعمل لها سينسز وتستد فعليا بحيث ان ده ممكن ده بيدي نوع من التراستنج للموديل اللي انا طلعته وبيخليني ممكن اخد عليها بعد كده؟
الهومولوجي موديلنج بيبقى دراسه بتستخدمها في كذا حاجه. الدراسه ممكن نستخدمها ان انا بدرس الاستراكشر كدراسه نفسه اللي انا ما عنديش غير السيكونس بتاعه وما عنديش فرصه ثانيه ادرس بيها. واخد بالك؟ فانا بيبقى عندي ثيوريتيكال ستادي عليه، بشوف شكل الريسبتور ممكن يكون ايه. لكن لما بستخدمه ان انا اعمل بيه داتا بيس سيرش زي الخطه المعموله، يبقى العاقبه بان انا بالمركبات اللي تطلع عندي بعملها تيستنج في الاخر. واخد حضرتك؟ يبقى الكونفيدنس ان انا بالاوتبوت لو انا هستخدمه في الداتا بيس سيرش. تمام؟ اما انا ممكن بستخدمه عشان اعمل دراسه دي، بستخدمه عشان يديني تصور ممكن اعمل ديزاين او اشوف ايه ادرس شكل الريسبتور ممكن يكون ايه. تمام؟ في ظل ان انا ما عنديش داتا اكسبيرمنت. تمام؟ لكن انا دلوقتي بستخدمه وبعمل بيه داتا بيس سيرش، فبالتالي التست اوف كونفيدنس اني في الاخر الاوتبوت بعمل له اكسبيرمنتال على الانزيم ان فيترو وعلى البكتيريا، ولما يديني الريزلت بقول له اوكي البروسيدور بتاعتي تمام.
شكرا دكتور عواد، متشكر جدا لحضرتك. ننتقل للدكتور علاء. دكتور علاء، بنرحب بحضرتك في بيتك الاساسي. دكتور علاء مش دكتور علاء مش موجود على اللاين. طيب اذا كان كده احنا بنشكر حضراتكم، ويعني نشكر المتحدثين الأكارم ولمحاضراتهم القيمة، ومرة أخرى بنرحب بكم وشكرا لكم وشكرا لكل الأودينس. السلام عليكم ورحمة الله.
وعليكم السلام ورحمة الله وبركاته.
السلام ورحمة الله وبركاته.